All right, let's get right into it. Welcome to today's explainer. We're launching a full-scale investigation into one of the most notorious and honestly fascinating, highly contagious equine diseases out there. We're going to unpack the science, the biology, and the sheer survival tactics of a microscopic enemy that's been plaguing horses for literally centuries. So, how exactly does a seemingly healthy horse trigger a massive disease outbreak? I mean, it sounds like the plot of a medical thriller, right? But it's actually the reality of the stealthy world of the Strangles transmission cycle. It's an absolute masterclass in pathogen survival. And by the end of this explainer, you'll see exactly how this biological mystery unfolds. Here's our roadmap for cracking the case today. We'll cover the threat of Strangles, meet the bacterial culprit, track the transmission cycle, uncover the hidden carrier state, look at diagnosing the disease, and finally, breaking the transmission chain. Section 1. The threat of Strangles, a highly contagious equine disease. Because, well, to defeat an enemy, we first have to understand exactly what we're fighting. Strangles is a highly contagious febrile disease in horses. It targets the upper respiratory tract and is characterized by abscessation of regional lymph nodes. Now, the physical toll this takes on a horse is just intense. We're talking severe fevers and massive discharging abscesses right around the throat and neck area. In fact, these abscesses can swell up so much they literally strangle the animal's airway, which, yeah, is exactly how the disease got its pretty menacing name. But the danger doesn't always stop at the throat. In about 1% of affected animals, the disease takes a terrifying turn into what vets call bastard strangles. This is a severe, life-threatening complication where those abscesses actually spread beyond the regional lymph nodes and develop in multiple organs across the entire body. It basically turns a localized respiratory infection into a total systemic nightmare. Section 2. Meet the bacterial culprit, unmasking Streptococcus equi. So, what exactly is capable of causing this level of devastation? Let's zoom in to the microscopic level. Here is the rapid-fire biological identity of our culprit, scientifically known as Streptococcus equi subspecies equi. It's a pyogenic or pus-forming bacteria. Under a microscope, you'll see gram-positive cocci arranged in these distinct chains. In the lab, they're catalase negative, produce a complete beta-hemolytic clearing pattern, and they fall into the Lancefield Group C classification based on their cell wall antigens. Short version, it's a highly specialized chain-linking menace. And this brilliantly illustrates why this bacteria is just so successful. It literally wears microscopic armor. S. equi possesses a polysaccharide capsule and a cell wall M protein that creates an anti-phagocytic effect. In plain English, this armor stops the bacteria from being engulfed and destroyed by the horse's natural immune cells. The white blood cells come rushing in to fight the infection, but they just bounce right off. This lets the bacteria multiply unchecked and form those massive abscesses. Section 3. The transmission cycle, how the pathogen spreads. Okay, we know what it looks like and how it defends itself, so let's track how this armored pathogen actually moves from horse to horse. The crucial thing here is tracing the bacteria's chronological journey. Step 1, an infected horse sheds purulent exudate. That's the highly infectious pus from discharging lymph node abscesses or nasal discharge. Step 2, this leads to massive environmental contamination. We're talking water troughs, fences, shared tack, maybe even a handler's hands. Step 3, a new susceptible horse is exposed to that contaminated environment. And finally, step 4, the bacteria colonize the upper respiratory tract of this brand new host. And just like that, the cycle begins again. It's absolutely critical to realize that the environment in Step 2 is heavily influenced by human management. High-risk assemblies, things like sales, shows, race courses, or just mixing unfamiliar young horses together drastically increase transmission. When we crowd vulnerable animals together in high-stress environments, we're basically rolling out the red carpet for this pathogen to jump from host to host. Section 4, the hidden carrier state, the invisible reservoir. Now, if Strangles is so obvious, how does it keep surprising us? Let's look at the real plot twist of this explainer. Check out this stark contrast. On the left, we've got the acute, symptomatic horse. Fever, discharging abscesses, a highly visible illness. You instantly know to isolate this animal, right? But over on the right, we have the chronic convalescent carrier. This horse looks like it's made a full recovery. Absolutely no symptoms. But inside, live bacteria are being secretly harbored. This seemingly cured horse is actually an invisible threat to literally every animal it meets. So where exactly is the bacteria hiding out? Right here, the guttural pouch. It's an anatomical extension of the eustachian tube in the horse's head, and it acts as the perfect safe house for S. equi to lay low long after the clinical disease has passed. These carrier animals act as a living epidemiological reservoir. They silently shed the bacteria right into water troughs and pastures, which can trigger massive new outbreaks months or even years later, completely out of nowhere. Section 5. Diagnosing the disease, identifying the infiltrator. Since we obviously can't always rely on visible symptoms, we have to look to the lab to expose this stealthy pathogen. It all starts with prompt swabbing of nasal discharge or exudate. Streptococci are super susceptible to drying out, so rapid transport is absolutely key. Once cultured aerobically at 37 degrees Celsius for 24 to 48 hours on blood agar, we start seeing specific visual laboratory markers. We're looking for mucoid colonies up to 4 millimeters wide, surrounded by a wide, clear zone of beta hemolysis. That means they've completely destroyed the red blood cells around them. When a lab sees this, it's the very first major warning sign of an outbreak. But scientists have to be incredibly precise here, because S. equi has some very close cousins, like S. zooepidemicus and S. equisimilis. They're all group C streptococci. This table breaks down the exact sugar fermentation profile needed to isolate our culprit. If you look at the S. equi column, it's uniquely negative for trehalose, sorbitol, and lactose, but positive for maltose. That specific biochemical fingerprint is exactly how we differentiate it from other, less dangerous organisms. But what about those hidden carrier animals that don't have any active discharge to swab? Well, that is where PCR detection comes in. Polymerase chain reaction technology is a highly sensitive molecular tool that detects the actual DNA of the organism. Because carriers show zero clinical signs, running a PCR on washes from the guttural pouch is absolutely vital for uncovering those hidden reservoirs before they can infect the rest of the herd. Section 6. Breaking the transmission chain, biosecurity and control. Now that we know how this pathogen operates, how it hides, and how to detect it, let's talk about how we apply this science to actually protect the herd. If you take just one actionable number away from this entire explainer, make it this one right here. 10 days. That is the strict minimum isolation period required for any newly introduced or even returning horses to a property. You simply have to give the disease time to reveal itself in a quarantine setting before you ever allow that horse to mix with your established, susceptible herd. And if an outbreak does happen, here is your crucial biosecurity checklist. First, isolate clinically suspect animals immediately, like at the very first sign of a fever. Second, avoid overcrowding and mixing age groups. Remember how human management fuels that transmission cycle? Third, implement rigorous cleaning and disinfection of all shared equipment and environments. And lastly, relentlessly identify and manage those carrier animals using the PCR tools we just talked about. While some inactivated vaccines do exist in certain countries, the literature notes their efficacy is kind of questionable. So these strict biosecurity steps, they're our true primary defense. Which leaves us with this final provocative thought. Are hidden carriers silently threatening your herd's biosecurity right now? The science clearly shows us that what we can't see is often the most dangerous threat of all. Thanks so much for joining me for this explainer. I hope this deep dive into the transmission cycle of Strangles empowers you to rethink your environmental risks and keep your animals safe. Until next time, stay curious and stay focused.